So thank you, Dennis. Um, so one thing I cannot see is the chat, but otherwise, what I'm looking at is my first slide uh, featuring myself and then the other people in my group right now, Caroline
Yeah.
Kinter and Brooke Loveland.
Yeah. We'll let Karen take care of the chat, and
Okay.
then she'll interrupt
No,
you, or
I'll-
so people interrupt you. Can we do that?
Yeah, I'll keep an eye on it.
Okay.
I'm seeing the same view that you are, Mike. So.
Okay, very good. So, so part of the reason why my talk, I've completely redone my talk. So I'll get this version to y'all. It's not a part of the current box. And the reason is this: we've been very fortunate since August to essentially completely retool our lab. And so, you know, a little bit of what and Dennis mentioned it yesterday. What I usually talk about is a technique called selected reaction monitoring, or SRM. But because of some new instruments, it's now PRM, or parallel reaction monitoring, which functionally is not that different, but semantically, it's one of those things that mass spectrometry people get very excited about. And so two new instruments in the lab: the Exploris 480 that we use for data independent acquisition that we've been hearing about, and then this new Stellar instrument that's what we use for parallel reaction monitoring. And the big step here is that we can now do several hundred proteins in a single run, whereas we used to only do 50 to 60, which once upon a time was a huge number, but just has gotten smaller and smaller as the size of the experiments have gotten bigger and bigger. And so I've retooled my talk to try and include more and more of this data. And so I'm going to touch on four things today. The first is to answer this question I think that sometimes comes up, and maybe is what is targeted quantitative proteomics? And then second off, where did the methods come from? And then farther down, we'll look a little bit at what the data look like, and then kind of finish up with how exactly do we work with people, and how do we do these experiments for people? And so the first question is what do we mean by targeted? And there's a question today about Western blot, and it's somewhat analogous to that, where people come to us to tell us, with the idea of what do you want to measure. And so, the unbiased experiments that Dennis and Rick and Sam have been talking about, and Karen have been talking about, these are the opposite of that. These are completely biased, that we go in and try and measure the things that you ask us to measure. And the inspiration for this, this desire can be validation of the unbiased proteomics data. So as you get your DIA data as an example, to try and validate some of that with additional analyses. We've had people to come to us with RNA-seq data and observations from their RNA-seq experiment, and want to measure a particular group of proteins based on that. And then I think this is a little bit underappreciated, but probably is the most of what we actually do, is that people come to us with a list of relevant proteins that they want to look at to test their hypothesis. And this kind of started with some collaborations that I had, that originally people thought their treatment created oxidative stress. And so we set up a panel of proteins to measure the oxidative stress enzymes. And then later somebody came to me wanting to look at the yeast kinetochore complex, and we put together a set of assays to measure those proteins. More recently, we had a lab come to us that they wanted to measure a protein called APEX1 and its companion DNA damage enzymes, and we set that up for them. And so, but these tend to be the two directions that experiments come to us. Once we have this inspiration, we then sit and develop specific methods to make sure those proteins get measured as well as they possibly can. And Dennis invoked this today, the idea of best flyers. We want to find a couple or three peptides that respond the best, so therefore will give us the best sensitivity for those proteins, and ideally the most specific measurement of them. We also use tryptic digestion, and so these proteins are going to be marker of that protein of interest. We have done a few experiments where people have come to us and want to be sure to be able to differentiate between a couple of isoforms, and that can push us to make sure that we include a couple of distinguishing peptides in our what I call an assay for a given protein. From there, we just develop a very fundamental quantitative LC-MS experiment where we selectively detect those best flyer peptides. And we take advantage of the mass spectrometry to give us sensitivity and specificity. And so this is just an example of a validation experiment. And you may have seen a little of this data yesterday. My lab likes to feed mice different types of fat diets, and this happens to be a fish oil diet, and then looking at the changes in protein expression that go on in the heart. For this picture, I drew a very tight kind of definition of what was increased and decreased, with 20% increase versus 20% decrease. You know, just a classic volcano plot. It was a relatively big experiment. We had 3 groups with n equals 12 in each. And so, you know, out of this, and, you know, Dennis was hovering over particular points and lighting them up to see what they might be, you know, would come this idea of then us perusing our data, looking at some of the results here, and, you know, perhaps generating a series of follow-up questions. And, you know, we see this across the board increase in some of these beta oxidation enzymes in the mitochondria. You know, could this all be correct? Are they really all increased expression? As we look at the peroxisomal forms of some of those, you know, these are increases in expression on the 4 to up to 16-fold increase. So, you know, they really that large an increase. And then in the middle of it all is this one peroxisomal protein that's going down. And so these are the sort of validation questions that would come out of an unbiased experiment. Excuse me. Alternatively, you know, we can pick up the textbook and put together a list of the mitochondrial and peroxisomal beta oxidation enzymes, and they're all shown here in blue. So this can be our wish list. This would be a hypothesis testing approach. As we look at this wish list, maybe some subsequent questions that would come up of, you know, we produce acetyl-CoA that goes off into the Krebs cycle. Does the Krebs cycle capacity increase to handle the flux of acetyl-CoA? And, you know, and this is all about lipid processing. Would glycolysis be affected? You know, so again, the idea of developing a list based on questions to test a specific hypothesis based on our knowledge of our biological system. The methods that we use, as I kind of implied, our main method is this parallel reaction monitoring. It's done on this stellar quadrupole linear ion trap instrument. We do, with the Exploris 480, do data independent acquisition experiments. And so actually re-interrogating those is an opportunity that might be out there. Rick implied this yesterday when he talked about the fact that there is primary data to go into these unbiased experiments to go in and look at. In these methods, just selected peptides. And these are peptides that we put together to want to be the best quantitative markers, so we don't just necessarily accept the coincidence of what the unbiased methods perhaps use. And so those peptides are selected. We work on this. It takes a little bit of an effort. And our PRM experiment, they're an innate part of the design of the MS method. We know the mass to charge ratio of the peptides that go on that list, and we know where they elute. And so it's a combination of we tell the mass spectrometer specifically what we want it to do, and at what point during our LC program do we want it to be doing that. And that helps us not only include more targets, but also enhances the specificity of the analysis, that we don't acquire superfluous data and expose ourselves to potential coincidental overlap with some things. When we're looking at data-independent data, data-independent acquisition data, we use what we learn here and what we set up here just to give what I call an enlightened reinterrogation of the data. We don't change the DIA data in any particular way. We just perhaps go in and look at it and plot some of it and evaluate it in an enlightened or educated manner. Again, these are all just fundamental quantitative liquid chromatography experiments. The chromatographic peak area is characteristic of the amount, and the role of the mass spectrometer, because of the way they operate, enhances the specificity because we have all these mass spectrometry characteristics that the detection has to satisfy. And then also mass spectrometers are inherently sensitive, so it enhances the sensitivity of the detection. And so I like to just kind of step back and remind everybody, and we touched a little on this yesterday, of how pretty mass spectrometry data can be for peptides. And this just happens to be one of our internal standard peptides. It's from horse serum albumin. This is the sequence of it. And kind of just the reminder is, we know the sequence of horse serum albumin. And so we can calculate, and we know the specificity of trypsin, so we can calculate the tryptic peptides that we would expect. And then for each of those tryptic peptides, we can carefully calculate what the mass spectrum should look like. And so this is the Y-ion series, starts with the C terminus of the peptide and extends outward. And so easy calculations to do. Rick and I used to use our slide rules to do this. Now computers do it in a microsecond. And now as we look at the CID spectrum, what we see is essentially the entire amino acid sequence of this peptide revealed in the CID spectrum. And these are the Y-ions. These are really good for parallel reaction monitoring. B-ions are not very useful. It would be the corresponding B-ion series goes from the N terminus outward, just because they all collapse down to B2 and A2 ions. So the high B ions don't really exist. And so, but you can imagine we now set up our mass spectrometer to fragment this particular peptide, and its mass to charge ratio of 575.3, fragment, and then look for fragment ions at 1036.5, 937.5, 823.4, and add all of these together and get a signal for that. Slightly unappreciated in all of this is, so the sequence ions here, these Y ions are determined by the amino acid sequence. The amino acid content allows us to calculate something called the hydrophobicity. And within our HPLC system, there's a very high correlation of the calculated hydrophobicity to the actual measured retention time. And so on top of being able to predict how the mass spectrometer is going to see it, we're also able to predict where it's going to show up in our HPLC chromatogram based on this correlation. And these are really good to, as you get your HPLC system calibrated, are really good to a couple or 3 minutes. And so it's a relatively modest time window. And so the result is this clear, confident detection of the peptides that we want. And, we have this predicted retention time. That's where we see the chromatographic peak. The different fragment ions are represented by the different colors. You can see the reproducibility of the HPLC. And this is an internal standard peptide. So the same amount was added to all of our samples. And so over a run of 24 samples, you can kind of see how reproducible all of that is. But, this generating this chromatogram is the goal of the development process. And so all of this works even in very complex samples. And so Dennis showed this sort of total ion current chromatogram. These kind of look like chromatographic peaks, and this is actually smooth to make it look prettier. But it's just a lot of data. And, essentially what we're doing, and this is a trypsin digest of a whole heart homogenate, is, these are two peptides that we would like to find. And we essentially ask the mass spectrometry system to show us first this series of fragment ions with this expectation of where they will be. And sure enough, we're able to see that chromatographic peak and know that we're detecting this particular peptide. And then the same for this other chromatographic peak. And all of it's based on this massive set of data that we're pulling out this very specific mass spectrometry characteristics that are determined by the amino acid sequence of the peptide. And so as well as this amino acid sequence relates, a peptide relates back to the parent protein, makes this a very specific assay for what's in this case acyl-CoA oxidase 1. And you see over here in the correlation where these two peptides show up. And so where do these methods come from? As I say, they're thoughtfully developed. And we spend a little time putting these together. They're based on the protein sequence and the LCMS characteristics. And so we have just a series of steps that we go through of gathering background information. There can be a little bit of communication problems of you may call your protein alpha ketoglutarate dehydrogenase, and I look it up and say, well, that's made of 3 different subunits. Do you want to measure them all? So there can be a little communication to figure out exactly what we want to measure. We can look at this thing called the BioGPS to figure out expression, give us some idea perhaps of maybe different tissues that might make the process easier. There's a peptide atlas out there that we can look at specific peptide detection. It's also very useful because it warns us about peptides that might be seen in more than one protein, and that can be quite useful and valuable. From there, what we do most often now is we probe some DIA data sets, and I just maintain a local data library of different tissues, different experiments that people submit to me to have a beginning resource to start to look and see what might be, which peptides might be our best flyers. We then begin to probe based on setting these things up, some representative samples with parallel reaction monitoring on our new instrument. And so we also have a sample library. We don't throw many things away until they're just dried up or gotten very old. And then as needed, we validate the proper detection with high resolution collision-induced dissociation on our Orbitrap instrument. And then the whole thing kind of collapses down to picking the 2 or 3 peptides that we like the best. And the things we like the best are based on a combination of high abundance, good peak shape. Some peptides just don't chromatograph well. Are there other closely eluting peaks that might complicate the analysis? And that's partly because, we are always leaning to the best. We always begin with the idea of the best flyers, but sometimes there's not much difference between the top 3 or 4 best flyers. And so part of our goal is also to make the data processing fast and clear. Just a little reminder that, this is what I mean by the bio GPS. Again, this is this protein ACOX-1 I'm kind of featuring throughout this. It's about 10 times more abundant in liver than it is in heart. And so we have access to liver tissue. And so that's where we would develop the method, and makes the method development go much more easily. The old way that we started was to look at the peptide atlas, and this just is a sense of what all is in there. These are the different peptides that have been linked by people to ACOX-1. The tendency is the best flyers are the ones that are seen the most often. Again, gives this idea of the calculated hydrophobicity. But there are, in the development process, and again, this is sometimes where I worry about the DIA results, is that we tend to avoid methionine because it can oxidize. We're very careful about cysteines because of the need to alkylate them. Tryptophans can oxidize. Deep peptides with DP bonds, these can cleave in certain acidic conditions. The CID spectra aren't as pretty or aren't as quite efficient if we have internal charge sites that make triply charged ions. And then, this highlights one that we probably wouldn't mess around with because it's very hydrophilic and therefore wouldn't stick. I don't really have any on here that seem too large, but the ones that are in bold would be a great starting point for trying to put a method together. And so just a little bit of thought at the beginning. And I kind of implied this earlier, that the key thing we need is a sample where they're clearly detectable. And most of our methods have been developed with endogenous amounts of proteins in favorable samples. Our new way has just been to go in and probe these DIA results. And so, this is the detection of these are the peptides that were seen in the DIA experiment in this ACOX-1 protein. And this kind of gives a nice graphical illustration of what would be the best flyer. So, all of these, I believe the trypsin does its job. So I believe there are equal amounts of all of these in our sample. And so, the difference between this NLQ peptide and this SFL peptide is it just responds better in the mass spectrometer. And mass spectroscopists are comfortable with this idea. There's a paper out there by a relatively prominent lab run by Ruby Aebersold that talks about the fact that the response for the best flyers is essentially peptide independent. And so it kind of takes away our need to calibrate all the best flyers, that the best flyers from ACOX-1 respond about the same as the best flyers from horse serum albumin. That's our internal standard. And so this gives us an attack to do absolute quantitation. And so, but this is a great starting point, and something, again, that we maintain through this data library. We can and have worked with low abundance proteins through a couple of different resources, either getting the overexpressed lysates or making synthetic peptides. So, harder peptides harder proteins are not impossible. And, this is just the final result. The programming for the mass spectrometer of what peptides to detect, what mass to charge ratio to fragment, the window which they elute. And then this also gets crunched over into our data analysis method, and we use Skyline to do that. And so, I'm kind of show lots of chromatograms. And so, it's a combination of the mass spectrometry, the liquid chromatography, and then this aha moment of seeing in a sample where we have nice clear detection. When we do, we pick our peptides, we enter all this into our local database, little quiet celebration that we have the method together, and now it goes on to our list of working assays. Just a key point here, sometimes people think this seems like a lot of work, but once it's developed, it's good forever. And again, heading off some questions from yesterday, that modified peptides are possible. We still need this aha moment, so there are some additional challenges in terms of the stoichiometry of the modification and whether or not some of those modification peptides are even detectable by the LCMS methods that we use. So what do the data look like? You know, Dennis you got a taste of this in the new proteomics world, large spreadsheets. And, you know, and it's a lot of data. And then behind all of that, there are also all these primary chromatograms. So things that can be looked at. Moderate to high abundance proteins, as I say here, make lovely and convincing pictures, the sort of thing that convince reviewers, convince PIs, etc. Lower abundance proteins are detected far better by the PRM experiment, and some of them may be far less impressive by DIA and perhaps even a little bit discouraging, until you remember that these programs now use pretty powerful computer stuff, neural networks, machine learning, and whatnot. And, you know, just now the reminder that these things see data and see chromatograms, you know, better than these human beings and their eyes. And so, so this is an example of kind of how we send results out. So first off, we add either bovine serum albumin to most samples, horse serum albumin to cultured cell samples, but we add it at a known rate. And so the essentially the response of the best flyer peptides for our internal standard tell us what 23 picomoles per 100 micrograms of total protein looks like. And then for all of our analytes, we measure a couple or three different peptides. We take the geo mean, we normalize that up to our internal standard, and we get an actual concentration. And so these things have real meaning and real values to them. From there, it's just classic quantitative analysis, means and standard deviations. A well-done experiment, the precision will be in the 10 to 20% range. We start to get nervous when we see lots of things over 30% of you know, what went wrong. And this happened to be an N equals 12 experiment, so we had great statistical power, good t-test, and to show statistically significant differences. And so ACOX-1 and catalase were too early on that we showed them going up, and yes, indeed, they do go up by, you know, very large fold change. And then the last, that one that seemed to be going down, you know, indeed it does go down significantly. And then lastly, just as an idea of dynamic range, actin is in all of these samples. We measure it in all of these samples. It's in at 125 picomoles per 100 micrograms. So it's about 2,000 fold more abundant, yet measured in the same experiment as, you know, our ACOX-1 or our catalase or our ACAA1. And so this thing will have 1,000 lines. I only show a couple of groups, so 24 of the 36 columns. This is what the primary data looks like. These are PRM experiments. These are the fish oil diet, so higher protein expression. You know, Western blot people, you know, see bands and see lines and you know, they mean something to them. LCMS people see chromatographic peaks, and these mean a lot to us. And you can see, you know, the illustration going from our controlled diet where there's lower expression, lower abundance peaks, up to our fish oil diet where there's higher expression and higher abundance peaks. But you know, here even, you know, very clear, very clean chromatographic peaks. DIA, you know, can take, like I said, you know, this is where we trust the computers. it's a little bit harder to look at it at some of our lower abundance experiments. So maybe just not quite as ideal. But for higher abundance things in the DIA, it's still quite perfect. And so this is out of our DIA data, plotting these two actin peptides. And so, it can be a standalone experiment. That's a lot of what we use are targeted for. The equivalence of a highly multiplexed and precise quantitative Western blot. But they can be used to validate results either by, completely new analyses or, in some cases, a simple, educated reevaluation of the DIA data. Some other summaries here in terms of the methods that we use, with this point that once we develop the methods, they're good forever, so our capabilities continue to grow with time. And so just finally, how do people work with us? And it really is a matter of getting in contact with us and start to talking about, what you want to measure and why you want to measure it. And helping us through the process of setting those methods up. Ultimately, we like to get homogenates from y'all. Usually we like we ask that you do a protein assay and send us a particular amount. A lot of times, quite frankly, we go ahead and repeat that assay. And it was mentioned yesterday, that's really a very inefficient part of our sample handling. It uses a fairly good amount of the homogenate. My lab still uses in-gel digestion and what we call short run STS page. So these are really only run about 1.5 centimeters. But it's valuable to us to be able to see these different samples and understand problems before we're set to go. And so it's a tool that we continue to use. And then so finally, again, this bragging about the new instruments that we have in the lab. At that point, I'm happy to answer any questions.
Mike?
Yes.
Any questions for Mike before I tell him? So the one thing you said there, said the DIA, like you got to remember, you said the DIA doesn't look as pretty. So remember, DIA, we're getting 5,000 or 6,000, IDs there in a single run.
Or 10.
Right. Or 10,000. And your PRM method, you're at 50 or whatever. So you got
we're up to 500.
Oh, sorry, 500. Just want to compare apples to apples there. But that's why you do the quantitation, or the targeting, is if you look at that DIA data and you're like, it says it's there, I've repeated it and everything, and then you go do that, and it's now this monolith of a peak, because you have basically let the mass spectrometer focus all of its filtering ability on just isolating that one guy or the handful of things that you're looking for there. So.
Correct. And the idea that you've kind of carefully selected the peptides that you want to look at, and that's a little bit of a mystery sometimes in DIA is what peptides did they use, and it's not to say one is better than the other. It's just different thought process and different goals between the two of them. I sometimes say with unbiased experiments, essentially you're walking into it saying, I don't know anything about my system. I'm looking for clues of what might be changing and essentially ideas for a new direction for the work. Sometimes with the targeted is exactly the opposite. You want to make sure that you measure these particular proteins as best they can be measured.
Gotcha. One question I have too is where did the teddy bear go, Mike?
So I cleaned up the home studio. I went for the more blank background. I also had to move a vacuum cleaner. So this used to be my home office. Now it is grandchildren's bedroom. So
That's great. All
I
right.
had a
Any other?
question too. Well, I've got a question. Are there programs out there that you can like put in your protein and it will give you what the best flyers are, or
I
do you
don't,
just have to do that based on?
I kind of feel like they're fundamentally something that you look at. There is a program though that's now designed. It's called PRM Conductor, and it's with this idea of utilizing DIA data as your lead in, and then processing that DIA data to help you manipulate and make the selection um, of the peptides that you want to look at. And it even has some higher level logic to try and make you not to disperse the peptides over the entire LC range of the experiment.
Have you used Conductor yet, Mike?
I haven't, only because I was so excited to just get our current methods from, so there's a little bit of mass spectrometry talk. To get our new instrument, we had to trade in our old instrument, and it actually goes first. So all of your capabilities, current capabilities, go out the door. And so I was very excited just to be able to move our current methods onto the new instrument and see them work really, really well. But I haven't had a chance to get into the PRM Conductor yet.
Yeah, I hear good things, but I hear good things from Thermo about a piece of software developed by Thermo and used by people who work for Thermo.
Thermo.
So yeah, but it's on my short list of things to do as well.
Yeah, it's an interesting idea, and the only thing I say in the meantime is, I've put together a lot of methods over the years. I would say we're up to 600 or 700 different methods, kind of in the can, ready to go. And so the development process is something that I may have said this before. I see it as a Sudoku puzzle. It's maybe a little bit of a chore, but also kind of keeps the brain going, and so it's not as much of a chore to me as to put some of those methods together.
You're saying you still use the slide rule.
I still use the slide rule, exactly.
Karen, was there another question online?
No questions online.
Okay. Yeah, we got one here.
Hi, Dr. Kinter, can you hear me?
Yes.
So I have a question about the hydrophobicity that you mentioned. Is there like a cutoff or a number that is a good for the hydrophobicity value?
So I tend to not use anything 10 or under. Again, it's just experience with my HPLC system. And then I don't necessarily have an upper cutoff that I think about, only because they tend to be really big peptides anyway, which I would kind of naturally be less excited about, just because the bigger peptides tend to have kind of complicated fragmentation. The fragmentation sometimes just putting the right amount of energy. It just, the chemistry of the fragmentation just seems to be a little bit out of line compared to everybody else. And so I don't necessarily have a top end. It's a little bit the same with the ones that tend to be too hydrophilic, so not elute too early, tend to be smaller peptides, and they often don't have a good amount of their spectra are too simple, and there's just not as much amino acid sequence information, and so there seems to be a little more opportunity for coincidental overlap.
Thank you. My another question is, in one of your data slides that I saw, there was a higher percent RST in control versus the sample. Is it because of the low expression or that's a normal, even though it's like within less than 20%, it was 15 and 11?
A lot of that, I think, just is, I mean, that's the analytical chemistry of just quantitative analysis. I don't know, trying to figure out what, so it's just, so in the spreadsheet,
Yeah, in the spreadsheet.
yeah, I think that just is normal variation. I mean, overall, our variations were, oh, so it was essentially the ACOX one. Yes, ACOX one and the controls was 24% variation. A lot of the others were, 10 to 15 to maybe 20%. And it just is absolutely a lower abundance of abundance proteins in the control.
So that might be the reason since we have like
Correct. And low abundance chromatograms, you have, you start to incorporate noise into the chromatographic peak, and so it, that's where we tend to see our larger variabilities.
Thank you. And last question is that, so what would be some of the challenges if we have to like shorter the gradient from the longer for this method?
So a shorter, faster gradient, I'm starting to look at that and see, but some of it has to do with the scheduling, that the more there overlap there is in the elution of peptides, the more the mass spectrometer has to be doing at that moment in time. And so the shorter it's able to dwell on any particular signal. And so, and in fact, if I were to want to, absolutely be sure to measure a particular protein, I might include it in an assay with fewer other proteins to give the mass spectrometer the chance to dwell, accumulate, observe signal for as long as possible.
Thank you.
Guys ready to let Mike off the hook? Thank you, Mike, for